let's move on now to the next phase of T cell development, or what we can call the education of T cells. So the first phase of T cell development really had everything to do with making a T cell receptor. And it occurred in the thymus, and the whole goal was to produce a receptor that would be able to bind antigen, but it was completely irrespective to the actual specificity of antigen. So really just to put together two chains that can make a functional T cell receptor. Now, the second phase is going to be all about examining those T cell receptors and ensuring that A, they are able to work with self cells by recognizing MHC, but be able to not recognize self MHC so strongly or antigen associated with MHC that could be self antigen. This will include only looking at alpha beta T cells, because alpha beta T cells are the only subset of T cells that are restricted to MHC molecules. Gamma delta T cells don't recognize MHC, so we don't have to go through this testing of them. Yeah, as far as we know, when a gamma delta T cell rearranges its genes and is able to make a gamma delta T cell, it's done, and it can go on and look for antigen as far as that is understood at this point. But alpha beta has to go through 2 rounds of testing, and the first one is called positive selection, and the second one is called negative selection. We saw these two in the B cell development as well. We looked at them in reverse order. But with positive selection, that will occur first, and it ensures that T cells are able to recognize self-MHC. Negative selection is the same as what we saw with negative selection of B cells, where any T cell that recognizes self-antigen or binds too strongly to MHC will be removed from circulation. So gene rearrangements provide a huge amount, a large repertoire of T cell receptors. And then they could potentially recognize thousands and thousands and thousands of different types of MHC isoforms because of the different ones out in the human population. But only a small subset of the T cells that are made are going to have receptors that can recognize self-MHC. So T cells have to be positively selected for those that can recognize self-MHC isoforms, but not too strongly. If they don't recognize self-MHC, those T cells are never going to be able to interact with antigen, because antigen recognition with T cells must involve recognition of MHC as well. So positive selection is the process that ensures that this can happen. MHC molecules used in positive selection are going to self-assemble with self-peptides. So this is all happening in the thymus, and so we're looking at epithelial cells can have the MHC on their surface, and they're going to be displaying both MHC class 1 and MHC class 2, and they will be expressing self-peptides. So those thymic chordal epithelial cells are going to be responsible for interacting during positive selection with the developing T cells. And at this point, recall that the T cells have both CD4 and CD8 on their surface. And remember that CD4 recognizes MHC class 2 and CD8 recognizes MHC class 1. And so the epithelial cell can make contact with a double positive, either CD4 or CD8, and it's just whichever one they bind to that's going to determine which class that T cell is going to be restricted to. And we use the term MHC restriction to talk about how T cells can only recognize antigen in context of an MHC, and they're going to be restricted to the class that they interact with first. I think I have a slide that describes that coming up. If not, we'll mention it again. But this image kind of shows here that here we have an MHC class 1 molecule. And so our T cell receptor is going to bind, but also CD8 will. And this will go on to become a CD8 positive T cell. Down here, we have an MHC class 2 molecule. We have our T cell receptor interacting with it, but we also have CD4 interacting. And so then this would go on to become a CD4 positive T cell. So that recognition needs to happen in order for the cell to survive. If there's no recognition of those MHC molecules, there's no point in keeping those T cells around because they're never going to be able to interact with antigen because they don't recognize self-MHC. They may recognize the neighbor's MHC, depending on, like what the antigens or what the specificity is. So that's what we're talking about when we say MHC restriction. There's a lot of words on this slide, and I apologize for that. But it's kind of goes back to when we talked about B cells, and if a light chain wasn't, if the combination of the light chain and the heavy chain recognize self-antigen, the light chain could go back and try to edit its receptor. It could rearrange those genes just slightly different to get a little bit different antigen specificity. The same thing can happen with T cells, receptor editing, but this time the alpha chain is going to change just ever so slightly because it's going to be similar to that light chain in B cells. And so if that first alpha chain rearrangement on the pre-T cell interacts with self-antigen, then it will be, the reg enzymes will be signaled to turn off because we want not self-antigen, self-MHC, because we want a T cell that can recognize self-MHC. If the T cell does not bind self-MHC, then the alpha chain can go back, go through receptor editing, change out that alpha gene chain just slightly, and hopefully try again and be able to recognize MHC. So yeah, that's kind of what that's saying. The alpha chain is not subject to allelic exclusion the same way the beta chain is. And this is where I had mentioned how in the last lecture, how the beta chain is going to be the same in all those cells that proliferate from the one clone that's made after that first race. But the alpha chain isn't always going to be the same in a cell. And so you could potentially have T cells that express two different alpha chains because the alpha chains are rearranging at the same time. So on any one T cell, you might have different antigen specificities. The beta chain is the same, but the alpha chain might be different on receptors on the same cell. So it's not full allelic exclusion. Okay, now positive selection. Not only will select, this is good, this is the picture I want to show you, will not only select for a T cell receptor's ability to recognize self-antigen, but it also determines whether a T cell will become a CD4 or a CD8 positive. And I kind of drew that on that other slide. But again, if a T cell interacts, and these are double positive, right? So they have CD4 and CD8 on their surface. If a T cell interacts and recognizes self-MHC in its class 1, the T cell is destined to become a CD8 positive T cell. If the T cell interacts with antigen in conjunction with MHC class 2, then CD4 receptor is the one that's binding, and then that cell will end up becoming a CD4 positive T cell. And at this point, it's going to shed the T cell or the co-receptor that it isn't selected for and become a single positive cell. Okay, now negative selection is going to delete T cells whose antigen receptors bind too strongly to MHC molecules. Because yes, we need the T cell to recognize self because antigen is presented on a self protein, but it can't bind too strongly because then potentially that T cell could become autoreactive. Negative selection is going to be facilitated by a dendritic cell, but of the myeloid lineage. Myeloid lineage, meaning derived from the bone marrow. So hematopoietic stem cell, you have to go back to that hematopoiesis diagram where we had the myeloid and lineage, the myeloid and lymphoid lineages breakdown. Off of that myeloid branch, dendritic cells were made. And those dendritic cells can then migrate into the thymus, and they will have both MHC1 and MHC2 on their surface, and they are going to interact with T cells and remove any T cells that are interacting too strongly, because we don't want to have an autoreactive T cell. Yeah, so the T cell can be then programmed for cell death. Okay, this is all great and wonderful if the dendritic cells and the epithelial cells, whatever cells are in the thymus, are able to express proteins that the T cell could encounter at any time during circulation. But that's not always the case, because dendritic cells are able to, yes, share the proteins that they produce. Yes, share the proteins that they process when they uptake antigen from the environment, whether it's through phagocytosis or through endocytosis. And yeah, that'll be great for negative selection for those antigens. But we need to extend that to potentially any self protein that could be in the body. So I think one of the most incredible things of the entire immune system is that there are a few cell types that reside in the thymus that have a transcription factor that gets turned on called AIR. It stands for autoimmune regulator. And this transcription factor allows a tiny subset of cells in the thymus to turn on any gene in the entire DNA and make any protein of the body. I mean, that is nuts. Remember that all cells with a nucleus have the same DNA, whether they're in your toe, your liver, or your thymus, wherever. But depending on which tissue those cells are in, that determines which genes are able to be turned on. But not for those cells that have AIR. They can actually create tissue-specific proteins and present them to T cells in the thymus to prevent autoimmune T cells from leaving the thymus. It's just absolutely mind-blowing to me that that we have this level of negative selection available. Like, I mean, evolution, it is crazy. So negative selection then in the thymus leads to a group of T cells then that have central tolerance. Their T cell repertoire that should be good at not recognizing self tissue before they're released from the thymus. But just like we saw with B cells, T cells also have a mechanism in place for peripheral tolerance. Not so much they don't become energic, kind of, but they can, there's these things called T regulatory cells that we'll look at here on the next slide. So a self-reactive T cell outside of the thymus that encounters a self-antigen, can receive signals to die by apoptosis. But also there is this another additional way for shutting down an autoimmune disease out in the periphery. And it's with a subset of CD4 positive T cells, and they're called regulatory T cells, or we can call them Tregs. They have T cell receptors that recognize self-antigen, and when they are stimulated, they will shut down the immune response. So rather than inducing a cell to die, they will produce cytokines that calm the immune response to prevent a self-reactive mechanism. I don't know what word I'm looking for, response. So it is a subset of CD, it's one of the effector T cells of the CD4 group. Not only do they express CD4 on their surface, so they interact with MHC class 2, but they also express CD25 on their surface. So sometimes people will call them CD5 positive cells. More often than not, we call them Tregs. And they have a transcriptional repressor protein called FOXP3. And so you'll also hear them referred to as FOX3P or FOXP3 positive cells. When they come into contact with self-antigen in conjunction with MHC class 2, rather than becoming activated and proliferating and becoming like major effector cells for the immune response, they actually respond by secreting secreting cytokines that will decrease the immune response. And so this is another major mechanism of peripheral tolerance, is the shutting down of the immune system by T regulatory cells or Tregs. Okay. We saw that only a few T cells actually make it through full development through the and training and selection through the thymus. Those that do survive end up leaving the thymus as what we call mature naive T cells. They're mature in that they have all the machinery they need and parts and pieces to recognize antigen, to recognize it in the context of either MHC1 or MHC2, but they have yet to do so. And that's where the naive comes in. So they have not been activated by antigen yet. They will recirculate through the tissues of the body, through the secondary lymphoid tissues, through the lymph fluid, through the blood, and just circulating all around and moving through all the different lymph nodes. And they live for a long time. In fact, they live for longer than B cells do. And they can be around for many, many years, which is great, especially if the thymus is starting to shut down. So you have a lot of these T cells early on in life. There are certain areas of the secondary lymphoid tissue that contain more T cells than other areas, and where they kind of hang out more often than not. And we call these T cell rich areas. Okay. They are going to interact then with B cells and end up becoming effector T cells once they engage with B cells and move on to being a mature effector T cells. We will dig into that in another chapter. But in short, that's when our CD8 positive T cells actually mature into our cytotoxic lymphocytes. And our CD4 positive T cells mature into either our T helper cells at this point or regulatory T cells. So now we really have 3 types of effector T cells that we've learned. We'll learn that there's more, but we'll start with that. So here's our summary slide. This kind of is of the whole development, phase 1 and 2, where we have the double negative T cells, progenitor cells coming in, rearranging their T cell receptor genes, end up becoming, for the most part, more often than not, an alpha beta T cell that's double positive. Positive selection will occur where we test the T cell's ability to bind self-MHC. Negative selection will remove those T cells that bind too tightly to self-protein in association with MHC. But this, and also positive selection will create a single positive T cell. And then they can leave and they can go out and they can search for antigen. As mature, MHC restricted, single positive, either CD4 or CD8 T cells. All right, that wraps up chapter 7, and we will, my mouse here, there we go, and we will, do some work with this and hopefully come out of this chapter with a pretty good understanding so that we can move into how the immune system works in our upcoming chapters.